All right, so we're currently working through this example of 3-point mapping using this data from Rollins Emerson, where he crossed these corn plants that had these traits of seed color, their leaf texture, and fertility. And in the first video, we talked through the information we needed to pull out of this setup, and then we answered the first of 5 questions. We decided that these genes were indeed linked. So now in this video, we're trying to figure out what are the parental allele combinations. That is, which of these alleles is grouped with the allele from the other gene on which chromosome. Okay. So we've talked about this in class. Let's walk through 2 approaches to figuring out what the parental allele combinations are. First, if we back up, you'll recall that in the question I told you about the parental generation, where we took a homozygous dominant plant and crossed it to a homozygous recessive plant. And in the F1, we got a heterozygous individual. From this information, just what I've shown you here, we know the allele combinations. So if we think it through, we can just say, well, what combination of alleles did this F1 organism get from this homozygous dominant parent? Well, if we think about that parent, that one parent gave a chromosome with the dominant alleles on it, with the uppercase V, the uppercase GL, and the uppercase VA. Well, the other parent had to give a chromosome with the recessive alleles, a lowercase v, lowercase gl, and a lowercase va. Because these alleles came grouped together from one parent or the other, we know exactly which of the alleles are linked to one another in this F1 organism. So we would say that the F1 genotype is this, where we're going to write down the genotype with this slash in the middle. The slash separates the 2 chromosomes. So on the left, we've got the alleles that are linked together on one chromosome. Again, this is the chromosome that was inherited by the F1 individual from the homozygous dominant parent. And then on the right side of the slash, we've got a chromosome that has these 3 alleles linked together on it. These 3 alleles were passed down on one chromosome that came from this homozygous recessive parent. So if we know about the parents that we generated an individual from, we may very well be able to figure out what the parental allele combinations are. In this case, all the dominant alleles on one chromosome, slash, and then all the recessive alleles on another chromosome. But there's another approach we can take. So let's say we didn't know about the parents, and I just told you that we crossed this heterozygous individual to this homozygous recessive individual. And then I just gave you the data from that cross. What did their offspring look like? From this data alone, we can determine what the parental allele combinations are. The way we can figure this out is that the parental allele combinations will be the most common in the offspring, because breaking apart the parental allele combinations requires crossing over between these genes. That's going to be less frequent because they're linked. Breaking them apart is going to be less common. So the most common outcome will be to keep the parental allele combinations intact. So to determine the parental allele combinations, all we have to do is look for the most common outcomes from the test cross. In this case, it's outcome 4 and 5, where we had 270 or 235 progeny, respectively. And these phenotypic classes represent the parental allele combinations. So here it just says, well, these ones are yellow, glossy, and variable, or green, rough, and normal. So how do we go from that to knowing what alleles are there? So at this point, it becomes important to write down the genotype that is associated with this phenotype. So I'm going to put back in here the table showing the symbols that I was using for each phenotype. So again, for seed color, where it's either green or yellow, green is dominant, yellow is recessive, we're using V's. For leaf texture, rough is dominant, glossy is recessive, and we're using the uppercase GL or lowercase gl. And then for fertility, normal is dominant, variable is recessive, and we're using the uppercase VA or lowercase VA. So if an organism is yellow, glossy, and variable, what alleles are together on this chromosome? At this point, it's important to remind you that we only have to consider what alleles came together from this first parent, the heterozygous parent. We already know what allele we got from the homozygous recessive parent. We got recessive alleles, right? So no matter what, from this parent, we got a recessive allele. That means which allele we get from the first parent determines the phenotype. If we get the dominant allele, we will see the dominant phenotype. If we get the recessive allele, we will see the recessive phenotype. Again, because the other parent is only donating recessive alleles. So again, this outcome, yellow, glossy, and variable, tells us which alleles we got from the first parent, and the fact that they were the most common says that those three alleles are together on one chromosome. They are linked. So which alleles are those? Yellow, glossy, and variable. Yellow, glossy, and variable are the recessive alleles. So we just say, well, the lowercase v, the lowercase gl, and the lowercase va are together on one chromosome. How about for the other one that is most common, the green, the rough, the normal? That's going to represent the other chromosome from our parent. Well, they have the green, rough, and normal fertility. They have all the dominant alleles. So they have the uppercase V, uppercase GL, uppercase VA. And so we can write down over here, again, this annotation where we separate the two chromosomes with a slash. We say one chromosome has the dominant alleles, the other chromosome has the recessive alleles. And we determine that simply from the data from the test cross. Again, the most common outcomes tell us the allele combinations that were present on the chromosomes in the parent fly. Right. So next, we want to determine what is the order of these three genes on the chromosome. Okay. We're going to figure that out again from this data. So we've determined that these genes are in fact linked, that they're on the same chromosome. We've determined what genes, which alleles are together on the parental chromosomes in this fly. And so I'm going to change the way we write the genotypes down to this way with the slash. Again, one chromosome has these dominant alleles on it. The other chromosome has the recessive alleles on it in this in this organism. And then in the other organism, again, it's homozygous recessive. So one chromosome has all recessive alleles, as does the other chromosome. All right, so what's the order of these genes on the chromosome? Okay. So there are three possible orders. Okay. One possible order is that the seed-colored gene, the V gene, is in the middle. And we could represent that in this way. We could say, well, GL is on the left, V is in the middle, and VA is on the right. For our purposes, this is exactly the same as having VA on the left, V in the middle, and GL on the right. All we're worried about in terms of the order is which gene is in the middle. It doesn't matter which end the other two genes are on, because it means the exact same thing. Okay. So that's one possibility. The seed color gene is in the middle, V is in the middle. Another possibility is that the fertility gene is in the middle, VA. So we could have GL-VA-V. We could have this order. Or the third possibility is that the leaf texture gene is in the middle. And we could have VA, GL, V in that order. And so we need to determine which of these three potential orders is correct. So the way we're going to do this is by looking at this outcome from the cross again. we can determine simply from the outcome of this cross what the gene order is. So the way we're going to do that is we're going to look at which outcomes were least common. The least common outcomes from the test cross represent what we call double crossovers. And these double crossovers tell us which gene is in the middle. So when you have the data from a cross, just find the two least common outcomes, and that will help you figure out which gene is in the middle. So how does that work? Well, all we have to do is to compare the double crossovers to the parental gene combinations. So we're going to compare genotypes. And then the one gene that is different is the one that's in the middle. So this is a rule that we're going to follow. So I'm going to remind you that we already figured out the parental gene combinations. Again, one of the parentals has all the recessive genes together. It's this one right here. And then the other parental has all the dominant genes together. It's this one right here. Okay. So in our parental allele combinations, we've got all dominants or all recessives. And then we've got these two outcomes that are the least represented. Those are our double crossovers. And we're going to compare them to the parents, and the one gene that's different from one of the parents will be the one that's in the middle. So here, for this yellow, rough, variable plant, we know that yellow is the recessive trait. We know that rough is the dominant trait, and that variable is the recessive trait. So I write down the gene or the allele combination that now exists in this plant. And I'll do that for the other one as well. Green, glossy, normal. Green is dominant, glossy is recessive, normal is dominant. And so now I'm going to compare this genotype that I got in the double recombinant to the parental. See, these two are mostly the same. They've both got little v, they've both got little va. But instead of lowercase gl, we've got the dominant uppercase GL allele. That tells us that the GL, the rough versus glossy leaf texture gene, is in the middle. We can confirm that by looking at the other pair here. So usually in the parent, we had the dominant alleles together. And now in this double crossover outcome, the GL allele has been switched. The uppercase V is still there, the uppercase VA is still there, but the middle one, the lowercase GL, is different. So we have just learned that this rough versus glossy allele, this leaf texture gene, is in the middle. So we would answer this question: what is the order of genes on the chromosome? This way. The gene order is, and you would write down either V-GL-VA, indicating that GL is in the middle, or another perfectly good answer is VA-GL-V. Again, GL is in the middle. All we care about is which one is in the middle. Okay, so far we've answered 3 of our questions. We've answered, are these genes linked on the same chromosome? We determined that in the first video. Then we figured out the parental allele combinations at the beginning of this video. And then the third question we answered was, which of those genes is in the middle? What is the order of the genes on the chromosomes? We just answered that. So I'm going to take a break here and then post a third video that answers the last of the questions we want to address when thinking about 3-point test crosses.